Gene therapy is an experimental technique that uses gene to treat or prevent disease. In the future, this technique may allow doctors to treat a disorder by inserting a gene into a patient's cells instead of using drugs or surgery. Researchers are testing several approaches to gene therapy, including replacing a mutated gene that causes disease with a healthy copy of the gene, inactivating or knocking out a mutated gene that is functioning improperly, and introducing a new gene into the body to help fight a disease. Cell therapy is the transfer of intact live cells into a patient to help lessen or cure a disease. The cells may originate from the patient or the autologous cells, or a donor or the allogenic cells. The cells used in the cell therapy can be classified by their potential to transform into a different cell types. Gene therapy is designed to introduce genetic material into cells to compensate for abnormal genes or to make a beneficial protein. If a mutated gene causes a necessary protein to be faulty or missing, gene therapy may be able to introduce a normal copy of the gene to restore the function of the protein.
The various strategies for gene therapy are the following. It requires to place a therapeutic gene, to put back or reinstall the virus-related gene using genetic data. The second strategy or another strategy to use in gene therapy is accompanied by the aided service of a healthy gene. The third strategy is to make natural cellular process into a medicine delivery, in a sense that this will be a special type of technology. The gene therapy utilizes genes to prescribe medicine to prevent disease. It heals or restores a faulty gene or attach a new gene within the attempt to enhance your body's ability to fight disease. That chromosome uses a fact-finding technique to deactivate a disease-causing gene that is not operating properly. 20 years ago, it was announced as a cure for everything from cancer to cystic fibrosis.
The most common type of cell therapy is by blood transfusion, and the transfusion of red blood cells, white blood cells, and platelets. Another common type of cell therapy is by transfusion of hematopoietic cell therapy or stem cell to create bone marrow, which has been present and performed for over 40 years. There are no currently formal and classification of stem cell therapy. Viruses as gene delivery vectors. Modified viruses are used as carriers in gene therapy. These viral vectors protect new gene from enzyme in the blood that can degrade it and deliver it into relevant cells. Viral vectors efficiently coerce to the cells to take up new gene to encode gene from virus particles and transport it usually to the brain nucleus.
The concept of gene therapy seems straightforward, but this is clearly an oversimplification, and numerous problems and risks exist that prevent gene therapy using viral vectors. Viruses can usually infect more than one type of cell. Thus, when viral vectors are used to carry genes into the body, they might infect healthy cells as well as cancer cells. Another danger is that the new gene might be inserted in the wrong location in the DNA, possibly causing harmful mutations in the DNA or even cancer. This has occurred in clinical trials for X-linked severe combined immunodeficiency patients in which hematopoietic stem cells were transduced with a corrective transgene using a retrovirus. And these led to the development of T-cell leukemia in 4 of 20 patients. In addition, when viruses are used to deliver DNA cells inside the patient's body, there is a slight chance that this DNA could unintentionally be introduced into the patient's reproductive cells. Theoretically, gene therapy benefits mankind as it allows us to discover treatment to diseases that are incurable nowadays. However, the actual application of such present number of possible risks that can alter the genetic engineering of those injected with it deems it to be impossible for now.
Supposedly, individuals ought to not utilize gene therapy to improve basic human characteristics such as tallness, insights, or athletic capacity, since gene therapy is utilized to repair genetic flaws and to avoid or cure genetic illness of an individual. Researchers and scientists are focusing on compensating for abnormal genes to make a beneficial protein. In order to help further the success rate of gene therapy and make it able to be widely used, first, scientists and researchers should focus on making the procedure safe before applying it to human patient. It will greatly help the improvement of gene therapy.